Well, hello, everyone, and welcome back to Science in the Age of COVID-19. Today, we are super happy to welcome Jacob Lemieux. This very well may be our last episode of this series, so we have saved the best for last. Lemieux meaning the best in French. Jake is a postdoc in the Sabeti lab and a practicing infectious disease physician at UMass General Hospital in Boston. His work is primarily focused on the various horrible things that are transmitted by tick bites. He also works on red blood cell parasites like malaria and also on respiratory viruses. And so the story he's going to tell us today is extremely fascinating. With SARS-CoV-2, of course, almost all the attention is paid on spike protein, but there are a lot of other open reading frames and probably non-coding RNAs and what have you in that genome. And he is trying to figure out what mutations in the genome are also super important. Okay, Jake.
Great. Well, thank you very much, Lauren, for that kind introduction. I'm really delighted to be here. I'm going to talk today about a recent preprint that we put out. I've actually finished my postdoc in Pardis's lab and have now established an independent group at Mass General Hospital, but continue to be closely involved at the Broad Institute and with the collaborative team. And in particular, I want to highlight that this work has been led by Fritz Obermeier, who's a really talented machine learning scientist at the Broad who works with our collaborative team. And it really comes out of longstanding efforts in the group to try to understand what are the genetic determinants driving microbial phenotypes, and how can we use large genomic data sets to identify those phenotypes? So I would invite you, if you're interested, to take a look at the preprint, which is on medRxiv. But I'd like to take a step back and really go to the beginning of the pandemic and recall the emergence of this mutation that changed in aspartic acid to a glycine at position 614 of the spike glycoprotein. This was one of the earliest signals of the emergence of novel variants that had increased transmissibility. And this emerged in January of 2020. And when it first appeared, it started to rise rapidly in frequency and prevalence in various populations. And we didn't know, was this something that was more transmissible, or was this simply a series of founder effects? And was hotly debated at the time, and to some extent actually still is. But it's become clear that there is a selective advantage to D614G, and this mutation has nearly swept to fixation and is present in almost 100% of circulating strains now, and really has been that way since the middle part of 2020. But this was really a foreshadowing of things to come, and history repeated itself. And in fact, has done so multiple times during the course of the pandemic. I'm showing here figures from early 2021 from Outbreak Info, showing the rise of B117 in the UK, where almost a year ago now exactly, it started to increase exponentially and then came to dominate by early 2021. And then a similar rise in South Africa of B1351. And the story of the last year, really, since the emergence of B117, is that of us as a world being blindsided by variants. And we only really seem to realize how problematic and transmissible these variants are once the horse is out of the barn, so to speak, once the variants have emerged and swept through multiple populations, we look back and kind of realize that the surges in cases have been due to highly transmissible variants, particularly B117 and B1617.2, or alpha and delta, respectively, in the WHO nomenclature. So how do we do better? I think many of us have realized that, well, the first thing is to produce the sequence data, the surveillance data, and that was a big challenge in the early part of the pandemic. But I think that problem has been successfully addressed in many places. So we have a lot of the data now, but we also need to kind of look at it and interpret it and make sense of it. And so I think many of us in the community have realized that it's important to kind of meet and look at the data and discuss it. And we have a meeting on Friday afternoons that I would welcome you to participate in if you're interested. But Friday, 3 p.m., we just kind of look at the variant surveillance data from the region and talk about it. And that's been really helpful. But it is an ad hoc approach. And it would be great if we had systematic, principled approaches to assess the emergence of new variants. And ideally, they would do three things. First, we would be able to determine which variants or lineages are more transmissible, and to do so in a quantitative way. Second, it would be great if we could do this not when the horse is out of the barn, but when the horse is either still in the barn or just emerging. And so can we predict transmissibility when a lineage first emerges, or even before or as it emerges? And this is a really challenging problem, in part because the data is most uncertain earliest after emergence. And finally, is there a way where we can identify transmissibility just from mutations alone, both to assist with the prediction, but also to provide a mechanistic understanding of what is driving transmissibility at a genetic level within the virus? So we set out to do that. And one of the things that we did was we took a look at the trajectories of variant frequency in the population and just kind of looked really hard at them and tried to understand the patterns that these variants tended to follow. And I think one of the observations that we made, and many other groups made this observation as well, is that there's initially an exponential increase in allele frequency of the variants. That exponential growth continues until the variant starts to reach closer to 100% of the population, and then it's sort of taken over from the other variants, and it's kind of reached its carrying capacity. And this is not unlike a chemical reaction or the introduction of a species into an environment. And this is well modeled by, or sort of really defines actually a logistic growth curve. And so this is a really valuable approach to understand these trajectories. But logistic growth curves are deterministic objects. And the challenge, of course, with natural populations is that there are multiple selective forces, there are multiple forces that influence evolution. The big ones that we have to worry about are selection, which is really what we would be modeling with logistic growth, and genetic drift, which is a stochastic force that is influenced strongly by population size. And there are other stochastic forces like founder effects and the linkage to other variants are sometimes called genetic draft, but drift is the major selective force. And the approach that we took leverages kind of two facts. One is that in large populations, selection dominates over drift. And by and large, viral populations during the SARS-CoV-2 pandemic have been quite large. And the other thing is that selection is a deterministic force which is systematic. And that if you look over many regions, the signal of the deterministic forces accumulates, whereas the signal of the random forces, like genetic drift, cancels out. So when you do that, if you make the assumption that something is growing in a large population, it's reasonable to fit a logistic growth model and to interpret the growth rate as actually the selection coefficient for the lineage that's growing. And when you look across multiple regions, if you see a systematic effect in the growth rate, that's a strong suggestion that a lineage is more transmissible relative to its peers, because the random effects, things like genetic drift, tend to average out over multiple regions. And you can see this, I think, quite beautifully if you look at the selective sweep that occurred with the Delta variant in our region here, as it did across the world. You can see consistent exponential growth leading to, well, consistent logistic growth leading to fixation in multiple states across the region. So logistic growth works well, but there is some behavior that it doesn't work that well for. And in particular, it's well models two lineage competitions where there's a much more transmissible lineage that outcompetes kind of everything else, or a collection of less transmissible lineages. But what do you do when you have multiple lineages and you have dynamics like what's shown at the right here? This is a true trajectory of B1526, which was New York regional lineage of concern that's kind of largely a fossil at this point, and is mostly of historical interest, but was more transmissible than what came before it, but was quickly outcompeted by yet more transmissible lineages like B117. So the binomial logistic growth model doesn't capture these multi-lineage dynamics well, and it actually fails quite badly on it. So the idea that we took, and I think really the credit for developing and implementing this model goes to Fritz, is to use a multi-lineage generalization of the binomial logistic regression model, which is called the multinomial logistic regression model. But I think the real innovation here is that we could actually build a gigantic hierarchical model in which we represent lineages as consisting of collections of mutations. So not only can we model the relative competition of lineages, but we can do it across different regions. So we can average over multiple regions, really extract the effect of selection, and also understand the particular mutations that are driving transmissibility. Now, this is a huge multi-level or hierarchical model. These models are most appropriately fit in a Bayesian context, but it has historically been difficult to fit large Bayesian multi-level models using traditional Bayesian methods to approximate the posterior, things like Markov chain Monte Carlo or Hamiltonian Monte Carlo. What Fritz and his colleagues developed or contributed to was a language called Pyro, which actually specializes in applying variational inference to approximating the posterior of these really complicated high-dimensional probability distributions. And using Pyro, we were able to fit this gigantic Bayesian hierarchical model. And it turns out that it kind of does what we hoped it would do and seems to do it really well. So the model structure is displayed in this figure. And what we do is we take an alignment of the raw data, in this case using GIS8 sequences, and we gratefully acknowledge all of the people around the world who contributed data to make this tremendous resource. And we cluster the sequence alignment, and in this case we use clustering provided by the Pango lineage classification system. And then we model the spatiotemporal dynamics of different lineages over time, regressing lineages on the mutations that comprise them. And from that large multi-level Bayesian model, we then extract relative transmissibility of each lineage, averaged over regions over time, as well as the relative transmissibility of mutations that make up individual lineages. We're taking advantage of convergent evolution that has resulted in mutations appearing in multiple lineages independently, such that we can learn mutation-specific effects using regression. And then we extract these coefficients, we fit the model, we extract the coefficients, we train the model, and then we make predictions about lineage dynamics and the relative transmissibility of mutations. So here is the second figure from our paper. And this is a display of the relative transmissibility of lineages. These are the lineage level estimates. This is a complicated figure, so I'm going to walk you through it. The size of each dot corresponds to the total number of cases from each lineage at the end of the training period, which was July 2021. The position on the x-axis is when each lineage emerged, and the y-axis corresponds to the relative transmissibility as compared to the original Wuhan lineage of the lineage in question. And what you can see is that there was a successive emergence of lineages for about the first year of the pandemic that really didn't have major differences in transmissibility. And then about a year ago, there was the systematic emergence of more transmissible lineages. And these lineages led to large surges in cases and came to dominate the pandemic. Lineages that you've heard of, like B117, P1, and 617.2. So what we can see is a successive emergence of more transmissible lineages that have dominated the pandemic. With the information about relative transmissibility of lineages, we can then use that information to survey for more transmissible lineages that may not have captured sufficient attention or may have concerning signals, and forecast into the future what lineages are going to predominate in which regions. Here is applying the model to data from England, and the model here is fit with data up through April of 2021. The end of the model fit, all the data for the model fit is generated with the filled circles, and the X's are forecasts. And so even with very little insight into the trajectory of B1617.2, the model correctly predicts, in this case, the rapid emergence of B1617.2 and the rapid disappearance of B117, which is, of course, what occurred. Now, this is not the only way one could have seen these dynamics. And in fact, there were many excellent models, some of which used binomial logistic regression and other methods to correctly forecast these dynamics. So certainly this isn't the only way that one could do this, and there are many ways that one could likely build a more accurate local model. But I think there's a couple advantages of having a global model like this one. One is that the model is systematically surveying in a way that's almost impossible for a human being to do every geographic region and looking for systematic differences in transmissibility. So there's a surveillance aspect to a global model that's really advantageous. But the second thing is that this model is borrowing data across regions. So this is taking to account the relative transmissibility of B1617.2, not just in the UK, but also in multiple other geographic regions around the world. That makes for more accurate and robust predictions. And that's one of the really nice features of a large model that incorporates multiple geographic regions into it. But I think the most exciting thing about this approach is that this model is hierarchical over the mutations that comprise a lineage. And so we're able to propagate the inference down to the level of the amino acid changes that make up each lineage. And so it yields a kind of nested genome-wide association study for the individual mutational events that may be promoting transmissibility. And it does this in an objective way that looks across the full length of the genome. So here is a Manhattan plot of the whole SARS-CoV-2 genome and the relative effect on transmissibility of each mutation that we model. What you can see is that the model recapitulates many of the findings, in particular the known importance of spike, especially regions within spike that are known to be linked to increased transmissibility. One of the strongest hits in the model is D614G, supporting the major role that this particular substitution has in promoting transmissibility. But we also see many familiar faces like N501Y and T, mutations in the receptor binding domain that are thought to increase binding to the ACE2 receptor. Other mutations like L452R and Q that are also thought or in some cases have been demonstrated to promote transmissibility. And things like T478K, which are present in multiple lineages, especially delta containing the delta lineages. There are two other hotspots in spike that the model identifies. One are substitutions and small deletions in the N-terminal domain, roughly in the 140 to 150 region. And then substitutions in the furin cleavage site, particularly those that confer additional positive charge. The P681 position, especially P681R and H. R in particular has been experimentally shown to confer increased transmissibility, are highlighted by the model. There's some other positions there that have been a little less well studied, 675H and 655Y. So I think the model is both recapitulating known biology in spike, but also highlighting new areas where there's a lot more to learn. Speaking of new areas where there's a lot more to learn, one of the strongest hits is in the nucleocapsid. There's a spike associated with increased transmissibility around position 200 of the nucleocapsid. And this does seem to be quite a strong signal that's really localized quite sharply around position 200. And we were sort of puzzled by, whether this was, what to make of this and how this might be working. And so we're very excited to see this preprint from Jennifer Doudna's lab, cited all that came out in medRxiv in I think July of 2021. And these substitutions in the linker region also caught their eye as a part of the nucleocapsid because they're in many or almost all of the variants of concern or interest. And what they did is they created this beautiful assay where they had the structural proteins of SARS-CoV-2 cloned into plasmids along with the packaging RNA that they identified so that they could create virus-like particles. And then they had a luciferase-based readout where they could assess the role of different mutations on viral packaging. And what they found, and I'm just showing their data from their preprint because I think it's relevant here, is that these mutations right around position 200 in the linker region between the N-terminal and C-terminal domains of the nucleocapsid seem to be experimentally associated with increased viral packaging and presence in purified viral particles in their assay. So I think it supports, there's still a lot more to learn, but it supports that this is a region where there's a lot more to learn and likely something interesting is going on. I think really highlighting the predictive value of our model for potentially highlighting areas of new biology or even identifying areas where we should look to understand new biology. Another example of that is Morph1A and 1B, which are vast polypeptides that are kind of whole genomes unto themselves and are processed to create a series of protein products. And these both had several strong signals of transmissibility in our analysis. And when we looked in more detail as to where the protein products occur along the larger polypeptide in relation to these signals of increased transmissibility, we found that particularly in NSP2 and NSP6, which are not well-studied non-structural proteins, there were strong signals promoting transmissibility in each of those places. So again, lots of really interesting places and potentially a really interesting biology to learn. And we think that the model is pointing the way, at least where we might start looking. One of the other things that this can do is we can try to build smaller models and say, okay, well, this is the model we can build with the whole genome, but could we do just as well by building a model, training a model just with the substitutions within spike protein or within the nucleocapsid? And that in turn allows us to infer the relative contribution of each protein or each open reading frame to transmissibility of the virus. And when we do this, as compared to the full model ranked by the evidence lower bound, which is just the variational version of the marginal likelihood, we see that the spike mutation, spike gene seems to, number one, doesn't recapitulate the model of the full genome, but it seems to have the strongest effect on transmissibility. And that is unsurprising and consistent with other results. However, this also highlights the important role of non-spike proteins that seem to have a major effect on transmissibility, particularly ORF1b and nucleocapsid. ORF3a and ORF8 also seem to be quite important. Finally, and I think in some ways, this is one of the most exciting features of the model, exploiting this fact of recurrent convergent evolution, we basically get to know these mutations in each of the lineages by observing the effect that individual mutations have on a lineage. And because we have so much data, and because there's so much convergent evolution, we can become familiar with the effect of these mutations on transmissibility. And in that way, we no longer need to wait for a lineage to spread throughout the population and to measure its trajectory. We can begin to make predictions about transmissibility from sequence alone. And so when we train a model without an observed lineage, just infer the mutation-specific coefficients without observing a given lineage, and then predict the relative transmissibility of each lineage with the leave one out model versus the full model, we find that actually the predictions are quite good. They're not perfect, but they're pretty good. And would imagine that as we observe more lineages, accumulate more data, and have a more accurate understanding of the relative contribution of individual mutations, these will even get better. I want to close by talking a little bit about some of the limitations of our approach, because I think it does a lot of cool things, but it does have some limitations that it's worth discussing. First, I want to note the double-edged sword of the approach to modeling relative transmissibility. The great benefit of modeling relative transmissibility is that it intrinsically accounts for a lot of the potential confounders, like the different rate of non-pharmaceutical interventions, or the different rate of vaccination, or other local factors that differ region by region. By comparing the relative competition of different lineages within a given region and learning a region-specific effect, which we then build into the larger model for a pooled effect, we're able to account for many of these confounders in a very kind of, I would say, elegant and useful way. But the caveat is that this assumes that all local forces affect variance equally. And I think that's largely true for non-pharmaceutical inventions like masking and social distancing, but may not be true, of course, for vaccines. I think we're still learning about this. And in many ways, the biggest effect seems to be between different platforms or actually waning of immunity over time. To a first order, it does seem that the vaccines work roughly equally against circulating variants, but there are some second order effects which we're not capturing in the model here. The other limitation is that we're not modeling the statistical interactions between mutations, in part because the space of pairwise and multi-wise interactions between mutations grows combinatorially. And so it isn't tractable to do that. At the same time, we know from the repeated emergence of patterns of mutations like ones in position 417, 484, and N50Y, that these mutations are not statistically independent and likely not functionally independent. Now, proof is in the pudding. This kind of simple, in a way, simple model performs quite well, but right now it doesn't include statistical interactions, and I think it would be quite difficult to incorporate them. It's also worth noting that the forecasts are quite good. As we looked at for England, it reliably predict 1 to 2 months into the future in places where we have good data and a good understanding of the lineages that are circulating. But the model can't account for the out of the blue emergence of highly transmissible variants that it hasn't seen yet. And so in places where B117 was exponentially increasing in frequency, that happened and in many cases reached fixation until 617.2 emerged. So there are limitations intrinsic to the forecasts related to the fact that every few months a highly transmissible lineage has emerged kind of out of the blue. So we can't just set this and forget it, but it does help us, I think, get a sense of the dynamics a few weeks, perhaps a little longer into the future, region by region. And finally, I just want to note that these predictions require experimental validation, and also just emphasize that right now it's a preprint, which is undergoing peer review. We would welcome any feedback, but these are inferences that require further characterization. So with that, I just want to summarize that we've built a large Bayesian hierarchical model over regions and over mutations to model the relative transmissibility of variants and the mutations that comprise them. We, as a result, produce an estimate of lineage by lineage relative transmissibility and mutation by mutation contributions to relative transmissibility. That provides quantitative comparisons of lineages that are circulating. It enables forecasts of lineage dynamics a few weeks, if not a little longer, into the future, and helps us guide experimental prioritization, and also highlights new regions, new areas of the virus that may be of functional relevance for transmissibility. And finally, it helps us get closer to sequence-based prediction of transmissibility, which will help us identify concerning lineages as they emerge rather than after they've emerged. So with that, I just want to thank the whole team, in particular Fritz, who has really been one of the driving forces behind this, and the I'd say brilliant programmer and implementer of Pyro models. So with that, I also want to just thank the whole team and acknowledge the submitters to GISAID and the funders who've supported the work, and point you to our preprint. So I'll take questions. Thank you very much.
So Jake, I was of course just kidding when I introduced you as a postdoc to see if you were paying attention. always deeply embarrassing, but
I am looking for postdocs.
That's what I meant to say.
If anybody is interested in doing a postdoc, please let me know, because we're just unpacking boxes here in the new lab. So
I know the feeling well. So that was great. Audience, please type your questions into the Q&A box and or raise your hand. And we've got a fellow expert on the line, Tyler. Can we get him unmuted?
Go ahead, Tyler. You should be able to ask your question.
Oh, awesome. Yeah, I was wondering, I guess it depends on how far a variant needs to be along to be quantified with your approach. But I was wondering to what extent you can see variants that do have signals of increased fitness, but they don't end up, quote unquote, making it, whether due to competition with other lineages, or what I'm super interested in is just stochasticity, like they start to grow exponentially, but just die out randomly. And so I'm wondering if you can see those lineages and learn a bit about how much of a fitness benefit really is needed in order to quote unquote make it.
Yeah, it's a great question. So I think part of it we can and do see. So in particular, if you look at figure 2 in the paper, there's all of these kind of unnamed intermediately more transmissible lineages that never went anywhere. It's the cloud of small dots behind the figure. So we can definitely identify and estimate the relative increased transmissibility of those lineages with confidence intervals, and that's all there, both in the paper and in the supplemental data for the paper. But what we haven't done, and I think we could do, but it would be difficult, is to quantify the relative contributions of basically selection versus drift. That's hard because the randomness is a function not only of sampling and of sequencing quality, but also of genetic drift, of sampling between generations of a population. But there's other random forces, like just the sampling for the sequencing programs, that differ quite a lot region by region. So I think it's something that we potentially could do, but it would be difficult. I think it's a great point. We don't understand why lineages die out. And then there are some lineages that are just also less transmissible than the original Wuhan strain. And there are some mutations that appear to make things less transmissible. You could imagine those being really interesting from a sort of druggability perspective. Those are potential vulnerabilities in the virus. But there's also kind of a circular thing where it's easier to measure things that promote transmissibility because you see more of them. So in our paper, we really kind of focused on the things that promoted transmissibility because we have a little more confidence in the data. We think those are easier to measure, but there is all of the things that reduce transmissibility, where there's some technical challenges, but the biology is likely to be really, really interesting.
You good, Tyler?
Yeah, that's great. Thanks.
Jake, I've got a similar question. So how easy would it be to feed metadata into the model and to figure out which mutations promote things other than transmissibility, say disease severity, male, female bias, et cetera, et cetera?
It's a great question. And that's something that we've been thinking a lot about. So not hard. And I think that transmissibility, we kind of got lucky because when you incorporate time and look at transmissibility over time, there's a natural interpretation of the growth rate in the logistic growth model as the selection coefficient. So there's sort of some natural biology that comes out of incorporating time explicitly as a covariate. But the general idea, you could, rather than growth rate over time, you could just look at the probability of having a severe infection and model that. The probability of having any other phenotype of interest. And so we think this is very doable. The metadata is hard to come by. There's geographic metadata, and I think that's what we were able to leverage to build a transmissibility model. But the clinical outcome metadata is not something that's publicly available. But because we're in a hospital network, we do have some of that, and we're beginning to bring that in. And for other people who would be interested in collaborating on that, we'd love to kind of work with you, because I think the more data, the better. There is another kind of cool aspect of this, which is genome-wide association studies in microbes have not been terribly successful compared to their human counterparts. And they're mostly good for identifying drug resistance loci, which have really strong phenotypic effects. But microbial variants that promote virulence and that kind of thing, they're hard to model in the traditional genome-wide association study framework. Microbial variation is just different. There tends to be a lot of it, tends to be quite geographically structured in pathogenicity islands and virulence plasmids. And there's a lot more convergent evolution. We see horizontal transfer of genes, and we see recurrent mutation of particular amino acid changes. So I think this is kind of a different approach or complementary approach to conducting microbial genome-wide association studies. And that's something that I'm really interested in exploring further.
That's a fascinating point. Sarada?
Okay, we have a question from Bernadetta. I hope I'm pronouncing your name right. I'm sorry if I'm not. You should be able to ask your question.
Yeah, I was just wondering, now that the sort of Delta has taken over all the other variants, do you have any predictions on which of the Delta AY lineages might have transmissibility advantage?
That's a great question. It's a really interesting point in the pandemic right now, just from a sort of lineage dynamic perspective, because we're kind of waiting to see what happens after Delta. I mean, right now Delta has essentially swept the world and is 100% of circulating viruses in the vast majority of places. So what is the heir to Delta, so to speak? And the Pango designations are helpful, but they're also confusing because there's just been this proliferation of essentially names. And they don't have a ton of genetic differences between them, but there are some. So it does take a little bit of pre-processing. We can't run the model every day right now, but we are running it periodically. Last week when we ran it, it did seem like AY29 had emerged as being, more transmissible, frankly, than Delta, not by a lot. The AY33 lineage has caught a lot of people's attention, including ours. It wasn't in the model when we ran it last week, but I do think that among the sublineages of Delta, at least by the Pango designation, AY33 is probably the one to pay attention to, and we'll see when we next run the model whether that, whether and to what extent that effect holds up. But I also want to note that, when 617.2 replaced B117, it wasn't a B117 derivative. It was something completely new. And so there do seem to be kind of quantum leaps in transmissibility that come from outside of subclades. So I think because there's so much B617, it's probably going to come from there, but I think we need to remain vigilant about the possibility that it will be outside the 617 clade. And we did use the model to estimate the differences in relative transmissibility between parents and their child lineages. And it's one of the supplemental figures in the paper. It's a really interesting distribution to look at because it's very heavy-tailed. And there are a handful of lineages, 617.2 and 621 in particular, that had quantum leaps and increased in their transmissibility from their parents. So still a lot to learn, and I think we need to continue to look widely for where the next variant of concern will come from.
Jake, I've got another question that I guess is not really a question, it's a comment. So you said that you thought it'd be super hard to introduce residue interactions into the model because, exponential growth, yada, yada. I don't think it will be. And so this is what I did my PhD on, was combinatorial optimization algorithms for, having in residues at each position, and coming up with a broad set of algorithms are called dead end elimination. And so let's talk offline, but you can, I think that there are ways to deterministically rule out various subclades and get the residue interactions in there. Because certainly for you showed E484K, I mean, that has shown, profound synergism with other nearby sites. I mean, this is what proteins do, right? So, I think you really need to get pairwise interactions at least in there. I mean, actually, proteins don't do pairwise interactions, they do n-wise interactions, but that I agree is probably impossible to model. But pairwise interactions, I will send you some papers, and I really think it shouldn't be too hard to get in there.
That's great, and I mean, I really appreciate that comment, and I agree with you. I mean, it's hard, but we probably can make some progress, in part because nature has already given us, some combinations that we should look at. And there are probably some combinations that we might have seen, but nature has not given us. And so, looking at the differences between what we observe and what we expect, just naively kind of counting the proportions of these may help us get somewhere. And then to try to begin to explore the combinatorial space, I mean, we have to. From a modeling perspective, it's hard, but as you say, I mean, that's the biology. I mean, these are real three-dimensional objects that flex and interact with each other.
Yeah. Hey, have you thought about feeding the model other sources of data, such as, for instance, the complete saturation mutagenesis studies from Jesse Bloom, for instance?
We have actually, first of all, I should say, Tyler, I appreciate your question, and your papers have been amazing, and we've actually pulled. I mean, it's great because the team and you provide the raw data, and so we did compare the mutational scan, the deep mutational scanning data for the ACE2 binding, and then also the antibody escape data. And we didn't see strong systematic relationships between transmissibility and binding or antibody escape. There were some weak relationships, sort of borderline significant. So sort of what we expected, it did seem like the RBD mutations were driven by binding, but I think there's multiple mechanisms, and we really don't even have a sense of what those are outside of RBD and outside of spike. But I do think those kinds of data sets are going to be really key to integrate with this type of modeling approach.
Yeah, no, I agree. I mean, you should just tell it everything you know. It can't hurt.
So,
Strada?
All right. No, I think I'm good with questions. Do we have any more audience questions? Audience, please type in your questions, raise your hand. All righty. Looks like it. Lauren, do you have any more questions?
Nope, I'm all asked out.
All right. So thanks, Jacob. That was a wonderful talk. So I guess we did save the best for the last. Speaking of which, as Lauren mentioned, this could will be the last talk in our series. So we'll take a moment to say a note of thanks. First of all, thanks to all our speakers, probably 50 plus of them, for sharing their what we call fresh off of the lab bench data. So go open science. Thanks to all our audience for joining us week after week, every week. Thanks also to you guys who join us to watch the recordings, because although we are still in the pandemic, life is resumed. You're all back at work. This is bang in the middle of the workday. So thanks for checking out our recordings. So you guys see Lauren and me every week, but there's actually a team who work with us to create the series and to curate the talks into a valuable resource. So time to thank them. Starting with our AV team, John. John is actually with us here today. Matt and Alex, who run the series and who also publish the recordings. Hazel and Ryan are our communication team members who actually broadcast this talk and curate the webpage. And last but not the least, Janine Stevens and Deborah Spurt, who have guided us along the way in so many different ways. So thanks to everyone. I hope we covered everyone. And if anybody from the audience would like to say a word, raise your hand. If not, Lauren, back to you.
Yeah, thanks to all our speakers and the people that helped. I know this definitely is pretty much the only thing that has kept me sane in the last 18 months. I'm really gonna miss it. And hopefully we've helped spread the good word of the latest research, and the recordings will live on. So thanks again, Jake. Thanks, John. Thanks, IT team. Thanks, HHMI.
Thanks for having me.
Thank you. Take care and be safe, everyone.
All right. See you on the other side.